Which has got banging Korean food, you say so. If you haven't been, you should go. Highly recommend really good Korean food.
It is.
It is. You know. What's the second?
He's a statistician, so.
So
Statistically, this Korean food.
So it got me making kimchi.
Really?
Yep.
You make kimchi?
I do.
That seems ambitious. How did the first batch of kimchi you ever made, how did that turn out?
Not bad.
Okay.
I used an apple instead of pear, and I think it tastes better with the pear.
Just make your entire kitchen stink though.
I don't think so. I don't think so. I didn't do the cabbage. I did the radish kimchi, which is good.
Yeah, and smell it when you walk in the house.
All right, let's see here. We are live streaming. For those who can't be here, YouTube. YouTube.
Smash that like and subscribe button.
Be sure to support us on Patreon.
Yeah.
Find me on YouTube.
All right. Yeah, I can't monetize because I don't have enough subscribers. I think you need at least 1,000.
What are we sitting at, 1?
But
I'm
I've got
supposed to
400,
can get you
400
like
some.
3,000 for just a couple $100.
Some of the how-to videos are probably the most popular ones
Oh, yeah.
that I get, like how to make a figure in Excel.
Right.
Yeah, a lot of like survivorship curve creations that gets a lot. But anyways, all right. So up in presentations, I've got papers and presentation topics. So I went through and came up with 13 different topics that you've got a choice from. And with each topic, I found basically a review article of some sort that kind of gives you a guide as to what we're talking about. So it's like evolution of sequential hermaphroditism. You know, it's like, what do you mean by that? Well, you check out Warner, 1975, and it goes into, you know, why you would do this, you know, what it is, why would you do it, what are the factors that favor it. These papers, I posted each of those papers up here, so you can check them out. Of course, they're not accessible, they're PDFs from journal articles. All right. But what I want you to do is to look through and come up with a ranking of your top 3 choices. All right. When you pick a topic, what we're going to do with our topics, when you pick a topic and we settle on when you're going to present, everyone in the class is expected to read the paper associated with that topic. So if you picked out the sequential hermaphroditism, I'm going to change that. I'm going to say theories of senescence, easier to say. All right. Bell, 1984. Everyone is expected to read that Bell 1984 paper before the presentation. All right. Your goal is to present the topic and explain the topic in terms of evolution and ecological theory. All right. What are the theory? What's the pros? What are the cons? What are things fighting against this? In some cases, why did it even evolve? All right. Are you just going to use this one paper? No. And the papers vary in size and length. Some of them are fairly short. Those you're going to probably spend a lot of time digging up extra resources. Some of them are fairly long and fairly complete, believe it or not. But you're probably going to have to look up some of the concepts, some of the ideas to learn it so that you can properly explain it. If somebody asks you a question in your topic, you are expected to have enough information where you can answer their question. All right. What are the topics? Evolution of sequential hermaphroditism. Evolutionary theories of senescence. We got a 1984 paper on that. Evolution of age and size at maturity. Evolution of offspring size and number. Good trade-off. Evolution of female choice in sexual selection. So it's not sexual selection per se, but it's female choice. What's driving that? And we will briefly talk about sexual selection, but this is the female choice. Why did that evolve? What's the benefit? What's the costs? All right. evolution of cooperation and altruism. We put them both together because they could evolve in a similar fashion. Evolution of dispersal, so leaving your natal area, moving. We have evolution of mutualism. We have evolution of virulence, so we're dealing with pathogens, and why would we start to exhibit some pretty significant pathology and so forth. We've got an interesting one, evolution of parasitism, right? Because we know it's evolved multiple times in multiple groups. So what are the factors driving that? And then we get to our plants, if you are interested in plants. Classification and evolution of plant breeding systems. So there are different breeding systems in plants. We have evolution of plant defenses. So you can think of plant herbivore interactions, why, what are the factors? What's that?
Sounds familiar.
Yeah.
Last semester seminar, plant animal
Was it?
interactions.
Oh, I thought you were.
I was gonna say biology.
No, so we're looking at more of the, we're taking the look at more of the plant responses, right? What are the effects of the plant? And in some cases, how do those defenses impact other aspects of its life and so forth? And then we had the evolution of parasitism. This was going to be focused more on just the animal world, because then we also have some parasitic plants, and that's also, we can just kind of focus in on the plants themselves and do the evolution of parasitism in plants. I think that might be an interesting topic. All right, so you're going to look through this. I'm going to need your top 3 choices by the end of the week, ranked in order. Number 1 is your top choice. Number 2, second choice. Number 3 is your third choice. I want to get these assigned by next week, Monday, so that way you can start working on your presentation. You're going to have a presentation. We're capping it at 30 minutes, so make sure you practice and you're in that 30-minute mark. The presentation will be shared. It will be shared with everyone, and we need some sort of like synopsis sheet or handout that kind of really gets everything down to the nitty. You could say to the nitty gritty, the key points of the presentation. All right, and then there will be questions from these topics on the final exam. So that's why you have to know it, you have to present it, and you have to be, you are going to be the expert on these. If you picked up the book, you will see that some of these are in the book. And I mean, there's actual chapter on age and size and maturity. I think also offspring size and number, plant breeding systems and so forth. But if you didn't pick up the book, since I didn't make it required, I'm providing these papers. And you'll see that the book references the papers. Minus, let's see here, this one, Parasitism in Plants, that's new. The book is like 2001 or 2003, I think, is the book. So anything past that is newer material. Maybe advancement. Any questions? All right. Email me. Or you can bring your topics to class on Wednesday. Any questions? Nope. We are good. I think these will be pretty interesting topics. I think hopefully you'll learn some stuff. It'll be interesting. Should be interesting. All right. Turn that off. So we are. Somebody erase the board. Pull out my chalk. What's that? I don't know. You tell me. Is ornithology this semester?
It's in this room.
Quite possibly.
It was today too. What it looks like to be your real place. Yeah. I don't know, it's like day 2 of the semester, so.
That's a bird.
Yeah.
This is a feather.
There it is.
Covers their butt.
Birds have feathers.
Psych.
All right. You do the same thing. For people that need to join virtually, see how this is gonna work. And I can share. If everyone's here, I don't plan on live streaming after this. Get your lectures. All right, so we introduced what evolutionary ecology is. We are going to jump right in with more of the genetics aspects of it. You kind of have to have a pretty good understanding of variation and stuff to kind of get to these traits. All right, so some of this will be reviewed. I know some of this will be reviewed. And I don't know how much we'll get through today, or all of it, but we'll see. All right. So, modes of expression of variation. All right, so how do we express the genotypic variation that we have? Well, we break it down into the type of traits that we have. So, we can have discrete traits. Typically what you think of. Typically what you think of when we talk about variation in traits in and of itself. So our phenotypes for these traits are distinct. Phenotypes distinct and non-overlapping. So distinct and non-overlapping traits. We usually depict these as frequency tables or bar charts. Bar charts, bar plots, or graphs. All right. So these are the traits that we typically see when you're learning about Mendelian genetics. So they often are simple Mendelian traits. Easy, right? Often simple Mendelian traits. Could be controlled by a single locus. Some cases could have multiple loci that ultimately determine this trait, but it determines it in a discrete fashion. All right? If we happen to see multiple morphs in our population, then this could suggest a few different things. So when we go out to the trait and you see two different morphs of that trait, so you can say the hair color. But, well, no, hair color's not. I guess 10 fingers versus 12 fingers. Right? Polydactyly. And if you have multiple morphs in this population and you see it, what could that suggest? Well, oftentimes natural selection isn't strong enough to remove one of them. And the one that we're talking about would be the one that's less fit than the other. So maybe it's not strong enough to remove this bad morph, and it just persists because of drift. Right? So selection's trying to take it out, but drift rapidly moves it away from removal. Could represent, or could mean that both traits have equal fitness. I'd say that's probably the simplest explanation. Or we could have spatial or temporal heterogeneity. So in one place or at one time period, it may favor one morph, but then when the environment changes, it favors the other morph, and you just basically have this back and forth, because our environment changes. So this happens because, again, fitness is a product of our environment. When we have this, when we have these discrete traits, we can analyze them just using standard population genetic theory. So this is basically Hardy-Weinberg equations, the Hardy-Weinberg equations that are expanded to include mutation, drift, migration, selection, multiple alleles, all that stuff. Are we going to do this? No, we're not going to do any of that. All right, so that's the simple discrete traits. I would venture a guess to say that most of the traits that we're interested in are actually quantitative traits. What are quantitative traits?
Maybe like traits that add to each other,
Good.
like being not tall.
Okay.
Yeah, traits that are like on a sliding scale.
All right. So yeah, it's basically with these traits, we're not discrete. We're not so discrete. It's just like one or the other. All right? So with our quantitative traits, we're dealing with phenotype that varies along a continuum. So it varies along a continuum, which basically has also been given the name of polygenic traits. When we're working with quantitative traits, as illustrated as a histogram. All right, so with our histogram, we have our phenotypic value, PV. We have our histogram, where the y-axis represents the number of individuals that are expressing that phenotypic value. So yeah, you say height, quantitative trait, we could exist anywhere along here on that phenotypic value. And it's all controlled by our underlying genes. All right? So quantitative traits are determined by genes at multiple loci. Did you guys cover this? It's a molecular biology course. Did you guys talk about that? Is that in the fall?
No.
Maybe you covered it in genetics. I guess it kind of depends on where you took it. I don't know if we our class does that. But we're determined by alleles of multiple loci. Probably the easiest thing to visualize is the case where they're additive. So it can be additive contributions. So example that we can use is, what you can think of as a light switch. So we've got 5 loci, and we can either be up here or down here. So it's either on or it's off. All right? And each of these can be expressed either on or off. The end result represents the product. So if we go through and look at our histogram and the number of individuals that have it, if we're all off, we're down here. If we're all on, we're up here. And then it's just your basic kind of a multinomial distribution at that point, where most common scenario will be those where you have some combination of these on and some combination of these off. All right, so that's kind of the simplest one. Things aren't always simple because several of these traits are actually much more complicated. So it can be much more complicated in terms of their expression. So expression at one locus may depend on others. And etc. So all of those more complex types of systems, more complex types of inheritance patterns, they can also apply here. Even if we can talk about dominance. If one is dominant, then it kind of masks the expression of some other ones as well. All right, so many of our traits are actually quantitative in nature. And these include the ones that we can talk about. All right, so wing size. For insects, that's quantitative. Birth weights, it's a quantitative trait. So anything that basically follows this histogram or can be illustrated on a histogram is likely quantitative. Nice thing about these is that the histogram itself typically follows a normal distribution, at least in the population. And you might say, hold on, thought you said it was a multinomial distribution. It did. Once you get past a certain sample size, the binomial distribution converges to the normal. So it typically follows a normal distribution. We could use that as an approximation. And this is great because our statistical properties of this distribution are well known. So statistical properties are well known, and because of that, we can then use some standard statistical tests to analyze evolutionary theory. So statistical tests can be used to test evolutionary theory. All right, so if they typically follow a normal distribution, then we can describe them using your standard statistics. With standard stats. So I'm going to go back to one of these that we've described. Based on our population, we can have an average phenotypic value for our population, and we can estimate our variance or standard deviation of this trait in our population. Which is great because now you can go things and start to do your regular statistical tests. Compare one population at this time point and then compare it 10 years later. Did the average change? Did the variance change? And all that stuff. And you're just using regular standard statistical tests. All right. Some cases, we're not necessarily interested in just looking at one of these traits, but actually testing maybe two traits. Is one trait more likely to change in the face of what our perceived selective factor is than the other? All right, so one thing that we could do is not only compare location, but we could compare variability. So we can compare the variability of our traits just in a single population. And so with our question when we're doing this, we're basically asking a question like, which trait responds more to selection? So we'll say is more sensitive and how do we do that? Well, we can look at variance, but that could be a problem. Why would that be a problem if we utilize variance? Right, I know. Yeah, stats class, isn't it? Great change request. Great change request. Here's the issue. With our variance and standard deviations, we're working with units. So let's pick up something. Let's talk about, let's say, predator awareness. All right, that's the trait that we're interested in. We introduce a predator on a moth, all right, and we want to say we have two different quantitative traits. Let's say we have wing size, and then we have coloration, mottling, all right, both quantitative traits. So the modeling you could quantify as like percent, maybe like percent uniformity. You can quantify how, the pixels or whatever. All right. We're on two different scales. So while standard deviation and variance might be on like say a millimeter type scale, your modeling's on a completely different scale. So it doesn't mean a whole lot if we just compare the variance. So if we're going to do something like this, we should use some other measure that kind of standardizes against whatever the raw value is. Well, when we talk about is coefficient of variation, and we learn about that in our stats class, where in the examples that we use there, we compare weights and we compare lengths. Well, if the average weight is 100 grams, but the average length is only 5 millimeters, 5 centimeters, probably better for 100 grams, 5 centimeters, right? You look at the change and you say, wow, you've got a 10-gram change in weight across our population, but in that 5 centimeters, we're only looking at like a half centimeter change. You'd say, well, the half centimeter change is a whole lot smaller, but we're on a completely different scale. We're on a single-digit scale versus a 3-digit scale at that point. So our coefficient of variation allows us then to make these comparisons to see which trait is more sensitive to selection, which one would produce more of a change when we introduce that predator. Is it wing lengths? Are we going to start seeing more variation appear in our modeling so that we become more modeled or less modeled and so forth? All right. So without getting too much into the stats and all of the analysis stuff, all right, the key thing that I want you to remember is that if we have all things being equal, I have more that I could have put up, but all things being equal, those traits that have more genetic and phenotypic variation have a higher potential for evolutionary change. And you may have encountered this before, but if you haven't, you need to know it. So, we've got two different traits, all things being equal. They're going to respond the same way. Which one has the higher potential for evolutionary change or variation? Why? Why is this?
Because its extremes are further away from each other,
Okay,
I guess.
and why is that important?
Because if the most survivable phenotype or the fittest phenotype is on either of the extremes, then it could move the entire bell curve, I guess, in that direction.
And when would that happen?
When there's a selective pressure.
Okay, if I say these are a response, this is the result of a selection pressure already. This is what we see. What's that?
Yeah, when the environment changes.
When it changes.
Okay, yeah.
When the environment changes, our selection pressure is gonna change, and you're exactly right.
Yeah.
If it changes, we don't know which one's going to be favored. But the wider we are, the more likely we have that genetic variation, so at least the population can survive. It may take out a massive chunk of it, but we still have individuals that have a phenotype that has a positive fitness. It gets us through those genetic changes. So kind of an important point. We needed the variation. The more variation we have the higher potential for evolutionary change. It doesn't mean it'll be faster, but we have the potential for that evolutionary change. And that's one of the reasons why variation, genetic variation, is so important in our populations. All right. So we had discrete traits, we had quantitative traits, we have another type of trait. And I don't know if you've encountered this before. We have threshold traits. Threshold traits, fine. So threshold traits are these traits that look like they're discrete. They look like they're discrete, but they're actually quantitative in nature. All right, so the underlying inheritance is quantitative, but they look discrete because once you get past a certain point of the allelic pressure or the allelic value, then you transition to that second phenotype. All right, so these are traits. That take on discrete values. But they're controlled by alleles at multiple loci. We refer to those traits as the liability. Is the liability. The liability are the alleles of the quantitative trait on which the threshold is based. So in this threshold trait, our alleles will contribute some value to the allelic strength, or to some level of expression. All right, so we have strength is going to be on our x-axis here for it. We'll say a histogram. All right. We have an allelic strength, TH, allelic strength, and then we're going to put up a threshold. So if our allelic strength is below that threshold, we get phenotype number 1. If we're above that threshold, we get our phenotype number 2. So it's inherited, and it's expressed as in a quantitative trait, but we have to get past a certain threshold in order to switch to that second phenotype. So you can see this looks to be like a discrete trait. We see it as a discrete trait, phenotype 1 and phenotype 2, but it's controlled through quantitative traits and this allelic strength that has to pass this threshold. All right. On this type, on these traits, if we were to do selection experiments, so fruit fly experiments or whatever, to try to figure out your expression. So you take your parents, phenotype 1, phenotype 2, you mate them, you get your probably phenotype 1, and then when you mate those to produce your F2 generations, they generally follow Mendelian genetics. And for us, this is two phenotypes. Right? So phenotype 1, phenotype 2. So our F2 generations generally follow the Mendelian 3-to-1 pattern. However, our F2 back crosses, so taking our F2 offspring, back crossing them with our parental phenotypes, will produce something completely different. They don't follow. So if we take that, your 3-to-1, we'll say phenotype 1 is more than phenotype 2. If you cross that back to the phenotype 1 parent, you would have expected all your offspring to be phenotype 1. You might not get that. If you back cross it with phenotype 2, you should stay in that same 3-to-1 because your phenotype 2 should still produce your phenotype 2. And these, no, you should get, if you back cross it, you would get 1 to 2-to-1. No, it's still 3-to-1. Let me think about it. 3-to-1 still? Without doing a Punnett square. Recessive. Yeah, recessive with your recessives. Your recessives should still be recessive, all recessives. With that recessive parent, your dominant should all be dominant with that parent, but we start seeing, very different changes. This is typically this is why, have you guys worked with fruit flies? No, some people did. This is why you go to F2 and then you back cross. And when this back cross doesn't work properly, something strange is going on. Example of this. Any guesses?
I've been thinking about salmon this whole time, but it's probably not right.
Tasty.
This is cheating because I googled it, so I was having a hard time conceptualizing it.
Yeah.
But apparently, schizophrenia,
Oh,
yeah, is
cool.
yeah, special traits. They have a certain number of alleles. Check that box. Yeah, ZOPHRE. Yeah.
Schizophrenia. Yeah, split personalities. Hip dysplasia in German Shepherds. I would have said hip dysplasia is probably the most popular or the most well-known example, but I don't know, schizophrenia is well-known too. Running into it. All right. So threshold traits. Adds variation to our population. More variation. Sexually dimorphic traits. So we haven't really addressed different traits in males versus females. Now we are. So when we have these traits where they differ in the sexes, this can be a direct or indirect result. It could be directly caused by it or just a consequence of gender-specific patterns of natural selection. And again, sexual selection is just a specific part of natural selection. So could say pattern of sexual selection and natural selection, or just natural selection encompasses that. All right, so when we think of sexually dimorphic traits, especially those traits that differ between males and females, oftentimes we're looking at how does that trait favorable for one sex but not for another, all right? And then how does the other sex then develop their own traits to help them succeed at what they're doing. So for our example, just think about those traits that are good at competing for mates, all right? And those are probably different from the traits that are good for raising young. So now you can see if the male is the one that's competing for the female to acquire the mating, you're going to have traits that get selected for competitive ability and not so much for raising the young. Females aren't necessarily competing. Their selection pressure is all about raising the young. So now we get this dichotomous split in what our sexes look like because selection's pushing them down two different paths. Yep. All right. But we don't have to have reproduction as the driver, as the driving force for these dimorphic traits. It does not need to be reproductive traits. We don't need those reproductive traits. And we don't need them tied to reproductive success. And we don't need them tied to reproductive success. It could be linked to, let's say, foraging differences or foraging preferences. And some of their social behavior, right? Different. So social behavior between male and females may kind of give rise to foraging differences. So you can have traits that increase foraging success, but then you also have that counter selective force of avoiding predation while you're out foraging. All right? So are we having traits that favor foraging success? Or are those traits selected for predator avoidance? And you can kind of think again about what the roles are. All right, it's not necessarily about reproducing, it's about also raising your young. So perhaps if foraging is tied to behaviors raising the young, where we may need to have pretty high foraging success. In the sense that when we go out and forage, we're spending as little time as possible out there, which necessarily has less importance on predator avoidance. With predator avoidance, we'll say cryptic colorations. But if we're always out there because we don't really have the duties of raising the young, now we're exposed to predators a larger percentage of the time. Maybe now we start selecting for predator avoidance and less about foraging success because we're spending much more time out there foraging and so forth. So we've got this interesting dynamic that can be working. Many times with our traits, they tend to be sexually antagonistic. And I'm sure you've covered this. So if we have sexually antagonistic traits, trait is selected for in one sex, but not in the other. And usually, that trait is going to be a hindrance to that other sex. Alright, so we can talk about the tail length in birds, right? Longer tail length, whatever that bird example is, it's everywhere. Right? Easier for preying felines to capture it. Right? But that's good for the males because the longer the tail, more mates you get. How did they figure that out, by the way?
Cut the tails off and glued them on, or didn't glue them on.
Exactly. Where'd you learn that?
Yeah, I have an answer. in the PDF on the that third Carter and Cardle.
Yeah. That's I can't even remember what that paper was on.
It's on a lot of that thing is on a lot of things, but it's in there.
Yeah. Oh yeah. Yeah. It is. Oh, just wait. They've got lizards have this flap.
The dewlap?
The dewlap. You have to wonder how they figured out what gets selected for. Need a pretty in-depth animal protocol to adjust that thing. But yeah, you cut the tail off, you extend it. Yeah, classic. What else do you do? Right. Heck, we do it with humans. Right. Put a wig on people, make them go out in public and see how many taunts, how many flirts they get or whatever.
They also colored in the red-winged blackbird shoulder spot things. You probably know the technical term for those spots.
Shoulders.
Shoulder spots. That's what we're calling them.
Yeah.
I did assume that. Yes, I did assume that.
All right, so we have numerous examples of these things. One I am going to talk about is, I don't have it posted, because I don't really think you need to read it. I think we cover it pretty well here. You could look it up. Delft et al., 1996. They look at flower size and number. That's what they look at. And they're interested in how big the flower is, how many you produce. Because normally the bigger the flower, the more energy investment it is to produce that showy flower at the cost of producing your seeds. All right? And when you initially look at this, it's very, very sloppy across the phylogeny. It's hard to kind of look at phylogenetic trends here. But what Delft kind of did was take the flowers and break it down into animal pollinated flowers and wind pollinated flowers. And that then started to reveal some of the important differences. So with our animal pollinated flowers, pollinated flowers, it comes down to mating success for the individual flower. So these are, you've got separate male and separate female flowers. The male flowers, the success is judged by how many visits they get by that pollinator. All right, so just because you have an animal visit the flower doesn't mean your pollen's going to find a female. So the more visits that you can get, the more likely it is that your pollen will get to a female flower and pollinate it, and thus your genes get to the next generation. All right? So in these male plants, the flower is selected for showiness. Showiness, that's a technical term, right? Kind of how elaborate it is, how bright it is. It's selected for reward, so giving a little bit of nectar or giving a lot of nectar for it. You're looking at overall basically attraction. So any trait that favors attraction, that's what's being selected for with the male flowers. Female flowers, it's different because now your reproductive success can happen just by a single visit. Because you're getting a pollinator. If that pollinator has pollen, it only takes one visit and then you get pollinated. So for our females, our success occurs in one visit. At least potentially. That's the theory. All right, so you get one visit and then you have seed set. So our selective force isn't really going to produce these attractive flowers, these showy flowers, increase reward. It doesn't have to. Instead, it only needs to invest enough to be good enough, be showy enough, just to get that one visit. And thus, you leave the rest of our energy for the offspring. So you can do seed size, you can do seed number. Anything where instead you're not expending energy to be showy and being attractive, you're going to be able to, you're only spending enough to be good enough to get in a visit, and then all the rest goes to your offspring. And that follows animal pollinated flowers, or at least that's the pattern for animal pollinated. The wind pollinated didn't follow this pattern, and hopefully you can kind of see why. We're not reliant on attracting a pollinator. We're relying on the wind. All right? So if we're relying on the wind, then the expectation is that natural selection will favor those flowers that have exposure of stamen and anthers to try to increase release of the pollen, increase dispersal of the pollen, and at the same way, catching the pollen as it moves by. All right, so we would limit petal and sepals and thus expose these stamen and anther and make pollination more likely. Is that what they saw? Nope, not at all. You probably read the paper. No, you didn't. That was Connor and Harlow. Did they get this? No, they didn't. And that was kind of actually interesting. So they didn't see this at all. Which happens. Do an experiment, didn't turn out as you expected. Now it's your job to figure out why. Right? What's going on? So what they started to kind of at least hypothesize is that perhaps the petals and sepals provide some other benefit to the flower. And that benefit may be in the form of protection. So if we're going to be good wind pollinators, you'd think you'd have a reduction in the size of the petals and the flowers so that we can increase pollination, but that didn't happen. So I think that these petals and sepals may actually provide some sort of protection for the developing sex organs, which then leads to this idea then that if the benefits from the protection exceeds the cost of being less likely for wind pollination, then it's going to persist, as long as we can still get some pollination to occur. So we have the protection. Yes, exposure to wind. So everything's about the costs and the benefits. If the benefits exceed the cost, we're going to continue doing that. And again, we still have some pollination happening. But we didn't see our trend. We didn't see this shift towards decreased size in the sepals and petals. All right. Is that how long we're gonna go? All right. So we have causes of variation and their evolutionary consequences. All right. So we have variation within our individual. Write it out of this chalk. When we talk about variation within our individual, we're actually talking about individualistic variation and not between individuals in our population. All right, so we're dealing basically in a change of phenotype in a single individual. Now there's two types of this variation, individualistic variation. All right, where we will see this change in our individual itself. We have that ontogenic variation. And we have somatic mutation. So it kind of gives us a change in our phenotype. I'll say with the ontogenic, I spent some time in the last years trying to figure out, is it ontogenic or ontogenetic? Yeah, we're going with ontogenic. I think you'll also see ontogenetic.
I'm saying it's not a tomato situation. 2 letters different. I'm sorry.
It's more like a color color.
Yeah. Program.
Yeah, exactly.
All right, we're going to start with this ontogenic variation. Ontogenic variation is this variation in time, and it is usually tied to age or developmental stage. So age or developmental stage. So these are phenotypes that'll change as we age. All right, and in many cases, these are programmed. So we're going to express them in two ways. We're going to express it in two ways. We've got age-related changes in our individual's phenotype. All right, our example, growth rates, hormone levels, etc., etc. So our growth rate start out, having a pretty steady growth rate, and then growth rate declines over time to the point where it stops. That is an ontogenic variation. The phenotypic trait that we're looking at would be growth rate. That growth rate changes through time. Hormone levels. If we restrict it to reproductive hormones, those change as we age. It changes through time. All right? So that's way number one. The second way that it gets expressed is that it changes in sequentially produced organs. So now we're dealing not so much with changes as we age, but it's tied to development. Changes in phenotype of sequentially produced organs. This one's kind of hard to come up with examples, but we have examples where fruit that is produced early on have more seeds than late producing fruit, or late produced fruit. So our phenotypic trait would be the number of seeds in our fruit, which again, normally would follow a normal distribution, but as it ages, as it's producing more and more fruit, those fruits that are produced later on in life tend to have fewer seeds. And I'm going to say it's not universal across the board, but this is something best example would be, again, organs, they're sequentially produced. Just keep on producing. All right? So detection and measurement of these traits, of these ontogenic traits, can actually be somewhat difficult. Yeah, we'll get this up. Of variation. All right, so it's going to be affected by both our genetic component and the temporal component. So we're talking about, we're trying to detect these changes and try to come up with summaries and synopsis of these things. Our genetic component could vary between individuals. And it's not just in the phenotypic trait, but also in our expression of that temporal component. All right. So what we're going to do is pause here today because there are some topics I want you to research and have answers for our next class. I believe we'll get to these in the next class. It'll be in the next, probably the next two classes. All right. So some of this may be review. Let's get these terms up here. So for next class. I want you to look up epistasis, epistatic variation. Probably have seen this before. I want you to look it up, just kind of review it, see, and kind of give an idea of how this might fit into the course. Cytoplasmic elements. again in terms cytoplasmic elements as they relate to phenotypic variation. Okay, got a couple more on here. And this one. The static variation. Epigenetic variation. I think that might be it. Yeah, I think that's probably all we're gonna need. I think so. I want you to look these things up. These things are related to additive versus non-additive genetic variation. So instead of me just kind of giving you the answers and defining these terms, I want you to look up these things and see how they affect our inheritance and stuff. And we'll discuss it. And if you find examples, that's awesome. We'll kind of get it into our notes. All right. So what we'll do, we'll pick up right where we left off here. Genetic versus temporal sources of variation. We're going to look at some figures that kind of look at that phenotypic value or the change in the phenotypic value through time, and see how we can predict whether ontogenic variation and or genetic variation is producing some of these differences. All right, and then once we finish those, then we will get to our basically inheritance, really kind of get in the background for selection. How is selection going to work? All right. I did have to record this lecture or live stream the lecture. Once I live stream it, it goes live on YouTube. I'll share the link in the playlist. I don't plan on live streaming and recording every day in class, just if we have anybody out for a reason. So if you run off to meetings and you're going to miss, maybe that's going to be the time I record. All right. All right. So I do have coupon books, and I left them. I walked out of the house and left them. So who wanted one? All right. I've got it. I think Taylor wanted one too. So I could take 20 bucks for cash, 20 bucks cash, or I could do Venmo or PayPal, but I have to look up those. So once I bring in the coupons, I'll give you the card, you give me the cash. All right? And actually, I flipped through it. Man, did I miss? They have got barbecue joints on there too. So they have some barbecue, Chick-fil-A, some fast food stuff. I'm thinking, oh my goodness,